Okay, so I just took the plates out of the incubator. I came back and this plate now is setting on top of the UV light table with the lid off. This is our untransformed on a regular plate, and it looks like regular bacteria. So this looks just like the control that we started with that I showed you in the first video. White. Okay, now we'll check out the glow. So we'll put this on, turn on the background, and I'll turn off the light so we can see it better. And we can see here that there is no glowing, so all of those are just regular white colonies with nothing glowing in there. So now we'll go ahead and turn this off. Lift this up, we'll take this plate off and we'll look at our second plate. So our second plate here is our untransformed on ampicillin. And as you recall, ampicillin is an antimicrobic. If we put it into our agar, then normal bacteria will not grow, and untransformed are normal bacteria because they do not have the plasmid with ampicillin resistant, and we see no growth on this plate as expected. Okay, so we don't really have to check it underneath the UV light, but I'll put that down, turn it on, and we don't see any bacteria there either because there are no bacteria. So we'll turn that off, lift that up. And we'll go to our next plate. Our next plate is plasmid P. So here's plasmid P. And if we look at this one, we see growth. So this is a positive plate. That means it has ampicillin, which means that normal bacteria cannot grow. So the only bacteria that grow are ampicillin resistant. We see only white colonies on this one. So no other colonies at all, no glowing colonies or anything. So this one worked really well. Plasmid P did a good job, and our transformation was successful in this plate. Now let's look at the glowing to see if anything glows. We'll turn that on, shut that down, and we'll turn off the light. And we see no glowing, so everything in there is white and not glowing for plasmid P. Okay, turn on the light. And now I'll go ahead and turn this off, open this up, and we'll look at our final plate. Our final plate is the plasmid that was plasmid A. So the bacteria transformed with plasmid A. And we can see here down in the bottom, so right down here, we can see two colonies that are green. The other colonies are white in there. So it grew on our on our ampicillin positive agar, which means that it does have ampicillin resistance. Okay, you'll notice that those green ones are dead center, and then there are little satellite colonies right around those green ones. Right, so a couple things. One is that those satellite colonies then could have the plasmid. It's also possible that we have very, very low ampicillin on the plate, that if those degrade the ampicillin around them, then some of the other cells could have grown enough that they actually got up and got a lot bigger. So now we'll shut this lid, turn on that, and we can see that those green ones are actually glowing. And we can see those nice glowing bacteria. Okay, so then if you think about how this relates to our study, go back and look at the table in the lab report of your predictions. Okay, just because this is glowing doesn't necessarily mean it's the bacterium that we want for our study. Right, so kind of look at those predictions, figure out, do we want plasmid A or do we want plasmid P for further analyses to degrade estrogen in the water. Now the cool thing about this is that we just make glowing bacteria. And by making glowing bacteria, that means we're about, halfway through the process of making a glowing monkey. A glowing, monkey that has babies that glow. And I guess, we could easily make a glowing person if we wanted to make a glowing person. So just for fun, I thought I'd give you a nice little close-up of the glowing colonies. So just remember this then. P is white and not glowing, and A is green and glowing for your lab report.